Okay, so we're going to be talking about the classical complement pathway of the complement system, okay? So the complement system, there's just some stuff you should know, okay? It's a part of the innate immune system. It is, okay? The complement proteins are always just in your blood ready to be activated, thus making it a part of your innate immune system, all right? So that being said, they compose of serum proteins, okay, which I just said. So these serum proteins will when you read about them, okay, it's gonna start with a C. It's pretty easy. So C is gonna start with a C, and you'll see, for example, C1, C2, and C3, okay, and C5 even, and that just all means they're complement proteins, okay? So the complement system actually has three pathways: the classical, the alternative, and the lectin, okay? They all differ. The classical pathway actually involves antibodies, okay, while the alternative and the lectin are antibody-independent pathways. So they all start with, they all have three similarities between all three of these pathways, okay? So they all start with activation of the C3, okay? And they all have a cascade, and they all end at the MAC. This is the most important here. They all end at the MAC. What does that mean? The MAC is a membrane attack complex, which actually pokes holes in the pathogen. So it all ends in poking holes in the pathogen with MAC, okay? All right, so the classical pathway. The classical pathway, like I said, the complement system is a part of the innate immune system, okay? Although the classical pathway involves antibodies, it's kind of integrated with adaptive immune system, all right? It's part of the innate immune system. So the classical pathway is activated by IgM and IgGs, okay? You need to know that, like memorize it. IgM and IgGs. Specifically, the IgGs involved are IgG1 and IgG3, okay? The IgM is the most efficient activator of the complement pathway. That being said, it needs a pathogen to be activated, okay? You can't just activate complement system without a pathogen, even if IgMs are in your blood, okay? Okay, so it all starts with a pathogen. The pathogen is first covered in antibodies, okay? And this is why I'm saying that adaptive is kind of integrated in the classical pathway, because it needs antibodies. And antibodies come from plasma cells. Plasma cells are B cells, right? And that's a part of the humoral. And humoral is a part of adaptive. So it's kind of integrated with adaptive here, okay? But it's still innate immune system, all right? So IgMs and IgGs come from plasma cells. They cover the pathogen. And then the pathogen will now attract attention of complement proteins, okay? So specifically, what complement protein? It's called C1, okay? Let me look for it. I lost it. Here it is. Okay, so the C1 complement protein is actually a mighty beast. It has three important parts to it, okay? C1q, C1r, and C1s. So the C1, okay, has these three parts: Q, R, and S. All right, the C1q actually is a six-headed region, okay? So for example, this is the C1, all this right here, all right? And if you look closely, the C1q has six heads. It's all this here. So it's all this here, 1, 2, 3, 4, 5, 6, okay? All of this. And then attached to that are two C1q, C1s, and two C1rs, okay? So if I read this, C1 is a complex molecule comprising a large six-headed recognition unit termed C1q, and two molecules each of C1r and C1s, the enzymatic units of the complex, okay? So C1q is for attachment and recognition, and C1r and C1s are enzymes, okay? Like this says here. Okay, so C1q is going to come and attach to the Fc region of the antibody here, okay? And so C1q, the binding of C1q to this Fc region will actually activate C1r, okay? So C1q binds antibody at the Fc region. This activates C1R, just the sheer binding, okay? Activates C1R. C1R then cleaves C1s and activates it. So this is all the work that C1R does. It literally just goes, okay, wake up C1s, and it activates C1s. So C1s does the work from here, okay? C1s will then cleave C4. Where does it get the C4 from? In the serum, all right? It's just floating around. It grabs it, all right? So C4 is cleaved by C1s, okay? C4a, it's like released, okay? It doesn't want that. C4b is stayed attached to the pathogen, okay? So C4b stays. See, it's right here. It just hangs out. It just stays there, okay? C4b will now grab C2, all right? Where does it grab C2 from? The serum, all right? It grabs the complement protein C2, okay? Then it's going to present that C2 to C1s, okay? C1s will then cleave C2, okay? It's going to release C2b, all right? And C2a will actually now stay behind and hang out on top of the pathogen, okay? So C4b from before and C2a from now will actually join up together and create a new little complex called C4b2a, all right? We have a special name for this new complex. So remember, C4b stayed behind, and C2a stayed behind to hang out on top of the pathogen, and now they make a complex. So this new complex is called C3 convertase, okay? C3 convertase, as the name shows, okay, it's no surprise, it cleaves C3, okay? C3 convertase will cleave C3, C3a is released, C3b attaches, okay, to the C4b2a complex on the surface of the pathogen, okay? Okay, so this is what was on the surface before, and now C3b attaches there. And so it makes a new complex called C4b2a3b. Awesome. They also have a cool name for this one, right? No surprise. What is this one called? C5 convertase, okay? So this is what it looks like. Look at it hanging out up there, all right? So C5, so it grabs C5 from the serum, okay? And now C5 is going to be cleaved by a part of this complex, okay? The 2A part, all right? So the C5A is released when it's cleaved, and the C5B attaches to the complex. All right, so this complex is just getting bigger and bigger with each cleavage, okay? But it's still hanging out on top of the pathogen. So the new name for the complex with C5b involved is C4b2a3b, okay? So this is the last. Cleavage of C5 is the final enzymatic step in the classical pathway. Yay, because I could not keep track, all right? Right? Okay, good. Okay, what does that mean? So C5b, which is now associated with the complex, okay, hangs out onto the surface of the pathogen. C5b will then now grab C6 and C7 from the serum, okay? The cascade is still going, kind of, all right? C6 and the C7 will also join up with C8 and C9. All of this, C5b, 6, and 7, plus C8 and C9 is the membrane attack complex. It looks confusing when it's all written out, but look at this pretty picture I have here. All right, C5, that was the end of the enzyme reactions, right? Okay, there's no more enzyme reactions. What just happens now is C5b is just going to pick up these complement proteins from the serum. C5-6, C5-7, C5-8, and why do I keep saying C5? C6, C7, C8, and C9, okay? So it's going to grab all these, okay? And all these, C5 and all these put together is the MAC, the membrane attack complex. Remember, the membrane attack complex can now poke holes in the pathogen, okay? Thus killing it. Awesome. Perfect. We're done.